In this experiment, we are going to look at the triple sugar iron or TSI Agar. This is going to be day 1, which is going to be the setup, and this is activity 5-21. So the way that this would work is that each student would be given a TSI slant. And the TSI slant is going to have what's called a butt, which is this bottom part, and it has a slant. And so each person would be assigned one of these 7 organisms. So we have Pseudomonas aeruginosa, we have Alkaligenes faecalis, we have Escherichia coli, we have Salmonella choleraceus, we have Proteus mirabilis, Proteus vulgaris, and Proteus alcofacans. And so each person who has a tube would label it with their organism initials. Notice that there's 2 PAs, both Pseudomonas aeruginosa and Proteus alcofacans. So we would have those of you that were given Proteus alcofacans, you would label that PAL. That's how we would distinguish that from Pseudomonas aeruginosa. And so what we would do is each person would get 1 TSI slant and their organism. Now, when you inoculate a TSI slant, there are 2 things that you have to do. First, you need to use a needle, and you need to use the needle to stab inoculate your organism. So straight down, straight back up. So you're going to stab the butt. That's the bottom part, about 1 centimeter from the bottom. Then you're going to use your loop and zigzag inoculate your slant. And after you make your tube, you have to put the cap on loosely and then take your tube and put it in the incubator. And they would grow for 48 hours at 37 degrees Celsius. And so this would be our setup for our TSI Agar. And so I have a video demonstrating the technique of how to inoculate this tube. So in this experiment, we are looking at the triple sugar iron agar, TSI for short. So this is our TSI test. And so we have our triple sugar iron agar, and you'll notice that it's a slant, but it also has this region down here that we call the butt. So we have a slant and we have a butt. When we inoculate these, we are going to stab the butt, which basically means we're going to stab inoculate with a needle, just straight down and straight up. And then we will also inoculate with the loop and do a zigzag slant on the slant portion. So for this experiment, we would have 7 different organisms that we would test. Again, I'm just going to demonstrate 1. I am going to demonstrate E. coli. Again, your tubes would all be well labeled so that when you have 7 of these, you'd be able to tell what is what. So what I'm going to do is I'm going to start with the needle, and I'm going to do my stab inoculation first. So I take my needle, and I'm flaming it, and then I'm going to let it cool. I'm going to take my slant that has the bacteria, flame it, go in, pick up some bacteria, flame it, cap back on. Now, I'm going to stab inoculate this. Now, normally I would do this by the flame, but to make it easier to see, so this is my slant that I'm inoculating. I'm going to flame it, and then I'm going to stab inoculate. Straight down, about 1 centimeter from the bottom, and straight back up. Flame. Take my needle. Done with my needle. Take my loop. Okay, I'm going to let that cool. So I've already done the stab inoculation. Now I need to use my loop to do my zigzag inoculation on the slant. So I'm letting my loop cool. And then I'm going to take the cap. So again, I'm going to pick up bacteria from the same tube, flame it, go in, pick up some bacteria, flame it, So now I'm going to do my zigzag inoculation. Flame it. Remember, you always flame going in and out of the tube. So I'm going to start again at the base of the slant, and I zigzag up, flame it, cap, and then I'm going to flame sterilize my loop. Set that down. I would make all 7 of my organisms. Again, these are screw tops, so you're going to put the cap on tight and then a half turn loose so that the cap is still loose and air can get in. And so I would take all 7 of my tubes that I inoculate and I would stick them in the incubator, and after 48 hours I would record what my tubes look like. So now we're going to talk about our TSI day 2, talking about the readout. So the purpose of a TSI test is to differentiate bacteria on the basis of glucose fermentation, lactose fermentation, sucrose fermentation, and sulfur reduction. And so this test is many tests built into one. So we're testing for many different aspects. So when we look at this test, the name of the agar, the name of the media for your biochemical test, is simply triple sugar iron agar. That's the name of our media. And if we look at what is in the media, the first ingredient is going to be peptones. Peptones are there for general growth. So they're there to support general growth. Next, we have glucose. Notice that in this case, the glucose is 0.1% compared to lactose or sucrose, which are each 1%. Glucose is a monosaccharide. Lactose and sucrose are disaccharides. Lactose is glucose plus galactose. Sucrose is glucose plus fructose. So both of these are 2 sugars put together, whereas glucose is a monosaccharide. It's one single sugar. Now, if bacteria are given the choice, if they have a choice between peptones, glucose, or a disaccharide, if organisms can use glucose, that will be the preferred food source, meaning that's what they will use first if they have the ability to do so. So we have a low concentration of glucose as a substrate for fermentation to produce acids. So if the organism can ferment the glucose, they will ferment the glucose and they will produce acids. Now, once the glucose runs out, because it's only at a low concentration, bacteria then have another choice. They can either use a disaccharide like lactose or sucrose, or they can use peptones. And so the lactose and sucrose are there as a substrate, the disaccharide, for fermentation to produce acids. So we not only can test if the organism can ferment glucose, but we can also see if they can ferment lactose or sucrose in addition to glucose. So lactose and sucrose are there as our substrate, our disaccharide, for fermentation to produce acids. Next, we have ferrous ammonium sulfate. Ferrous ammonium sulfate is a hydrogen sulfide. H2S stands for hydrogen sulfide. Hydrogen sulfide detector reagent, meaning if H2S is present, this reagent is going to turn black. So if we see black, that means that the sulfur was reduced and produced H2S. Next, we have our sodium thiosulfate. This is our source of reducible sulfur, meaning it's a source of sulfur that bacteria could use to reduce sulfur into forming H2S, into forming hydrogen sulfide. Next, we have phenyl red. You've seen this many times. We've seen this in the urease test. We've seen this in the phenyl red broth test, etc. Phenyl red is our pH indicator. And so it's yellow when acidic, orange when neutral, pink red when alkaline. And so we can use the pH indicator, the phenyl red, to determine if the sugar was metabolized. And so this test has multiple parts. So when you're doing your biochemical sheet for the TSI, my recommendation would be to do one for the sulfur reduction and to do another one for the sugar metabolism. So for the sulfur reduction part, you have 2 substrates. We have our cysteine. Cysteine is going to be an amino acid substrate that is found in peptones. So those peptones are partially digested proteins, will contain the amino acid cysteine. So that is a substrate. That can be reduced, the sulfur can be reduced by an enzyme called cysteine desulfurase. And so cysteine desulfurase is an endoenzyme, because notice this amino acid substrate is small. So the enzyme is an endoenzyme, it's within the cell, and the enzyme is called, again, cysteine desulfurase. And what it's going to do is it's going to produce hydrogen sulfide. That's going to be our product. But in order to do that, it requires a low pH. So in order to form this H2S, one of the requirements is that you have a low pH. And the low pH comes from when the bacteria ferments the sugar. Because remember that if sugar is fermented, it's going to produce acids. The acids are going to lower the pH. So in order for this reaction to occur, we have to have a low pH. If we have a low pH and bacteria have this enzyme, they will produce this product. And the product is the hydrogen sulfide. H2S is hydrogen sulfide. So our hydrogen sulfide is our product that gets produced, and that is detected by the ferrous ammonium sulfate that is in the media. In this case, this reagent is not added after. This reagent is already in the media. So if H2S is present, it will react with the ferrous ammonium sulfate that's in the media, and it will turn black, because black is going to be positive for sulfur reduction, meaning that H2S was produced. Because again, the ferrous ammonium sulfate is going to detect the presence of H2S. How do we get H2S? Again, if bacteria can reduce the sulfur. So in this case, one of the substrates is going to be cysteine, and our enzyme is going to be cysteine desulfurase, and our product is H2S, hydrogen sulfide. Now, the cysteine is not the only source of sulfur in the media. The other source of sulfur in the media is our sodium thiosulfate, which I'll talk about in a minute. But the other thing I need to add is that for the negative test, meaning if sulfur is not reduced, the negative would be no black. So if we see no black, that means that sulfur was not reduced. No H2S was present or produced, which means that bacteria did not reduce the sulfur. Otherwise, we would have H2S. So back to our sodium thiosulfate. This is a substrate, and the substrate, remember, was found in the media. It was included in the media. And so again, this is our source of reducible sulfur. So here is our sodium thiosulfate. The enzyme in this case is called the thiosulfate reductase. That's the name of the enzyme. Again, this is an endoenzyme because the substrate is small. And I didn't include this on here because it's all the same at this point. The product is going to be the hydrogen sulfide. The ferrous ammonium sulfate is your H2S detector reagent. And if H2S is produced, it's going to turn black. And so this part, positive and negative, are the same for the top reaction and the bottom. And this also requires a low pH. So when you're writing your biochemical test, you want to include both substrates. So you need to include cysteine, which is your amino acid, and your sodium thiosulfate, which is in the media. Those are your two substrates. Your enzymes are going to be the cysteine desulfurase and thiosulfate reductase, both of which are endoenzymes. And the product for both, so this is where it all becomes the same, the product is going to be the hydrogen sulfide. The reagent is the ferrous ammonium sulfate, and positive would be black, because what that tells us is that sulfur was reduced and H2S was produced. And so this is your reaction. So you need to include on your biochemical sheet, there are two substrates, there are two enzymes, but there's only one product, there's only one reagent for this part. and you want to record what the positive looks like and the negative looks like for both reactions. So let's talk about what the different colors mean. So notice that when we started with our TSI agar, that the starting color was orange. And the starting color is orange because most bacteria are neutrophiles, meaning they grow best at a neutral pH. And so we want to start at the neutral pH, which, remember, with phenyl red would be orange. So our starting color is orange for our tube. Now, remember when we inoculated them, we both stabbed the butt, so stabbed down to the bottom, and we zigzagged on the slant. So when we start, bacteria have some choices for food. Remember that in the media, the food sources are going to be glucose, which is our monosaccharide, lactose or sucrose, which are our disaccharides, or peptones. So if bacteria are given the choice and they can use sugar, they will use glucose first. So if the organism is able to ferment the glucose, it's going to turn yellow. And so the tube is going to turn yellow, and it'll be yellow throughout at about 24 hours. So about 24 hours after incubation, if you were to check these, you would see yellow throughout if the organism was able to ferment the glucose. Now, if they cannot ferment the glucose, that means they're not going to be able to ferment the disaccharide either. So their other food choice would be peptones, using the proteins. And so if the bacteria use the peptones, remember that utilization of proteins is going to raise the pH. So the pH is going to go up and the tube is going to turn pink. So yellow is going to be that the sugars are used. Pink is going to be that the proteins are used. Now, if the organism utilizes the glucose, this test is done through completion until 48 hours. So at 24 hours, this test is not done. However, glucose is in a low concentration. So at about 24 hours, the glucose is going to run out. Now, when the glucose runs out, bacteria then have two choices for food. Either they can use the disaccharide, right? They can use the lactose or the sucrose, or they can use the protein. So when we are doing this experiment, bacteria will grow faster aerobically, right? So remember, we did a stab inoculation and we zigzagged on the slant. So if you think about where bacteria will grow faster, they will grow faster on the slant because when oxygen is available, bacteria can produce a higher ATP yield. They can get about 30 to 32 ATP per glucose compared to if they were to do fermentation. Fermentation only provides 2 ATP per glucose. So bacteria are going to grow faster on the slant than they are in the butt. Similarly, if you remember back to our standard plate count, when we mixed the bacteria with the agar and we poured it, remember that the colonies on the top of the agar were really large, whereas the colonies embedded in the agar were small. And I said that the reason that we see that difference in colony size, even though it was all E. coli, was that the bacteria that was growing aerobically, so on the top, grew faster than the bacteria growing within the agar. Same idea here. So in our TSI, we have a portion that is aerobic and a portion that is anaerobic. So the aerobic on the slant is where the bacteria grow faster. And what that means is they're going to run out of sugar faster, right? They're going to run out of their glucose before bacteria in the butt. Now, again, once they run out of the glucose on the slant, bacteria can then either use the disaccharide, so either the lactose or sucrose, or they can use the peptones. In this case, the butt will still stay yellow. The butt will still stay yellow because bacteria are growing slower in the butt, the bottom, and therefore they're not going to run out of their sugar in the bottom at 24 hours. So the bottom is going to stay yellow, but the slant is going to change color depending on bacteria's food source. So if the bacteria can use the disaccharide, either sucrose and or lactose, we can't say which one. It could be just sucrose, it could be just lactose, it could be both. We can't differentiate that. But if the tube stays yellow all throughout, the butt is yellow and the slant is yellow. What that means is when the glucose ran out, bacteria were then able to ferment the sucrose or lactose. And when they fermented the disaccharide, they continued to produce acids, which is why the phenyl red stayed yellow, because it's still acidic and the tube stays yellow. So if we see yellow all throughout the tube at 48 hours, that tells us that bacteria use glucose and at least one disaccharide. We don't know which one, but at least one disaccharide. However, not all bacteria can do that. Some bacteria, when they run out of glucose, they can't use a disaccharide. So now their other choice would be to use the protein. If bacteria use the protein or the peptones, that is going to raise the pH. And the pH is going to go up on the slant specifically, on the aerobic portion. Because remember that the bacteria are growing faster on the slant and therefore will run out of glucose faster. And when they run out of glucose, if they cannot use the disaccharide, they will switch to using the protein. So in this case, if you have a glucose-only fermenter, meaning they don't use a disaccharide, a glucose-only fermenter will have a yellow butt. This is the color of the butt. It'll be a yellow butt with a pink slant. That tells you it's a glucose-only fermenter. If at 48 hours the tube is yellow throughout, that means that they use glucose and at least one disaccharide, which is why they keep producing acids and the entire tube is yellow. So after the glucose runs out, we have two food choices. A disaccharide, so sucrose and or lactose, or proteins, which they will use aerobically on the slant because that's where the bacteria grows faster and that is where the glucose will run out faster. So this top part of this diagram is dealing with how bacteria use the sugars, right? And so remember that the purpose of a TSI is to differentiate bacteria on glucose fermentation, sucrose fermentation, lactose fermentation, and sulfur reduction. So this part that we've discussed does not yet look at the sulfur reduction. This is only looking at the sugar component, right? And so when you fill out your biochemical sheet, in addition to the one for the sulfur reduction, you want to have a second one that has the sugar information. And that is that the substrate would be glucose and or sucrose or lactose, right? Those would be our substrates in our test. And our enzyme would be various endoenzymes, right? This process, this fermentation is by various endoenzymes. And if the sugar is used, the product is going to be acidic. You're going to get acids, alcohol, plus or minus gases, right? Those are your products if the sugars are used. Under additional notes, you want to include the components about if it's yellow, right? If it's yellow, that tells you that the organism can use glucose and at least one disaccharide. If it's a yellow but a pink slant, that means that bacteria use glucose only and they do not use a disaccharide, so instead they will use the proteins. Or if the tube has no yellow and it's pink, either pink, meaning it's pink all throughout, that would mean that the bacteria use peptones aerobically and anaerobically. Or in some cases, you might see pink only on the slant, but not in the butt, meaning that the bacteria only use the proteins aerobically. So this is the sugar part of this pathway, right? This is what you would expect for these different parts. Now, we need to still talk about sulfur reduction. Remember that I said that for sulfur reduction, in order for sulfur reduction to occur, the pH must be acidic. So the pH must be acidic. So notice I'm not going to have a line coming down from this option because if there's no acids produced, if bacteria can't use the sugar, they also can't reduce the sulfur. So when we look at the sulfur reduction part, that has to be from this tube where the butt is going to be yellow. Because when the butt is yellow, when the bottom part is yellow, right? When the butt is yellow, that means that the pH is acidic, which also then will allow for hydrogen sulfide to be produced, That's when we get our H2S production. So if we look at this, right, we could have a slant that is pink and yellow. So the slant is pink and the butt is yellow. And the butt could also have black. And remember that the black is positive for sulfur reduction, meaning that the ferrous ammonium sulfate is going to detect the H2S that's produced. If it is a yellow slant and a yellow butt, it's yellow throughout, and it has black, then it also is positive for H2S. So if we see black, black is going to be positive for hydrogen sulfide production. And at least the bacteria had to use the glucose in order for the H2S to be produced. So you know that underneath the black is always going to be yellow because the pH has to be acidic in order for H2S to be produced, the hydrogen sulfide to be produced. So let's look at this. So here is the first type of tube we might get. And so it says slant is pink, and this has a K. K is referring to the alkaline pH. The butt is yellow. That is acidic. So you'll often see it written K/A. K/A means that the top is alkaline and the bottom is acidic. So if we see pink and yellow, what is our interpretation? And our interpretation is because the butt is yellow, that butt being yellow means that the bacteria had to ferment the glucose, right? The glucose was fermented. However, when the glucose ran out, the bacteria switched to peptone metabolism. Notice that the slant is pink. The slant becomes alkaline because bacteria did not use a disaccharide. They did not use the disaccharide. They used the peptones instead. So our interpretation for this is that this is a glucose-only fermenter. So this is a glucose-only fermenter. It's not able to use the disaccharide because if it was able to use the disaccharide, it would stay yellow throughout, but it's not. It's a glucose-only fermenter, and that's why we get a pH reversion on the slant. If I looked at that tube at 24 hours, it would have been yellow. However, we grew these for 48, and when we did that, that means that the top, remember the slant is going to grow faster, and when the slant grows faster, it's going to run out of glucose faster, and if it can't use a disaccharide, it's going to use the peptones, and the peptones are going to produce alkaline products, and it's going to raise the pH. So pink-yellow is a glucose-only fermenter. In our 7 example organisms that we tested, the organism that you would see this for would be our Proteus alcophaciens. So the Proteus alcophaciens is the one that's going to look like this. And so on the next slide, I have a picture that has all of our organisms and what they look like when they grow. And so you'll see that in just a minute. Here's another example. Notice this one is yellow throughout. And you can notice that the agar has actually separated. The agar has split. And the reason that happened, it's not that somebody did this test wrong. It's that gas was produced. Remember that when organisms ferment sugars, it can be plus or minus gases, meaning that they could produce gas. If they produce gas, like carbon dioxide, that gas that gets produced by the bacteria is going to cause the agar to separate. And so if you see these little air bubbles or air pockets in here, that's because the gas was produced. Now, yellow would be A over A, right? The top is acidic, the bottom is acidic. So did the bacteria use glucose? Yes, because the butt is yellow. Did they use a disaccharide? Yes, because the slant stayed yellow, which means that the bacteria continued to produce acids, which means they use at least one disaccharide. It could be that they use both, but it could be that they only have one. So yellow tells us that the organism ferments glucose and lactose and or sucrose. So it ferments at least one disaccharide. Our example organism for this would be Escherichia coli, E. coli. E. coli can ferment glucose and at least one disaccharide. We could have a situation where the slant is pink, the slant is alkaline, and the butt is neutral, it's orange. So if we have Kn, so neutral, what that means is that the organism uses peptones, but only aerobically. So this is a non-fermenter, does not use the sugar, but it uses the peptones aerobically. Notice that none of this is yellow, so sugar was not used. In this case, this organism is an obligate aerobe. It only grew and it only used the peptones aerobically. In our experiment, we don't have an organism that had this result, but you could. So I put Na there. We could end up with a situation where we get pink, meaning it's alkaline all throughout. What does that tell us? Again, it's a non-fermenter, but in addition to using peptones aerobically, they also use peptones anaerobically. So this organism is going to be non-saccharolytic. It did not use the sugar. It only used the peptones, both aerobically and anaerobically. And so in our experiment, our example of this would be our Pseudomonas aeruginosa and our Alkaligenes faecalis. These would be our examples of pink. Now we get to the ones that have black. So it has a yellow slant and a black butt. Do we know what color is under the black? And the answer is yes. It has to be acidic. So that's why I put an A here. Has to be acidic because it needs to be acidic for the hydrogen sulfide to be produced. So if it's yellow, again, that's an all sugar fermenter, meaning it uses glucose and at least one disaccharide. And it's positive for sulfur reduction. It also produced H2S. So our interpretation is that that's an H2S producer. So that's an organism that can reduce the sulfur. And it's an all sugar fermenter. It uses a monosaccharide and at least one disaccharide. Our example for this was our Proteus vulgaris. Proteus vulgaris gave us this result. Our last type of tube that we could see is where the slant is pink and the butt is black. Can underneath the butt be pink? Could that be a tube that was pink all throughout? And the answer is no. It had to be acidic in order for the black to occur. So what that means is the organism fermented the glucose because the butt had to be yellow, so it fermented the glucose, but it did not use a disaccharide because the slant turned pink. So this is positive for H2S production. This organism is able to reduce the sulfur, which is why we see black, and it's a glucose-only fermenter. Does not use a disaccharide because if it used a disaccharide, the slant would also be yellow, but the slant is not, it's pink. So that is our glucose-only fermenter. So for that one, the organism that we have would be Proteus mirabilis and Salmonella coloraceus. And so let's look at the actual examples for our tubes. So here are our results for the 7 organisms. So what we're seeing here is that here is Salmonella coloraceus. Notice you can see the yellow on the bottom. You can see the black, that's positive for H2S. The slant is going to be pink. And the slant that's pink tells us that this organism is a glucose-only fermenter. It only ferments the glucose, but it's able to produce H2S because it's black. The ferrous ammonium sulfate detected the H2S that was produced. So Salmonella coloraceus is positive for H2S, and it's a glucose-only fermenter. In the case of Proteus alcophaciens, notice that the bottom is yellow and the slant is pink. Again, this is a glucose-only fermenter. When the glucose ran out on the slant first, it then did pH reversion, meaning when the glucose ran out, this organism did not use a disaccharide. They only used the glucose, and when glucose ran out, they then switched to peptone metabolism. But it's negative for H2S. It's not black. Here we have Proteus vulgaris. Proteus vulgaris, we can see that the slant is going to be yellow, right? Our slant is going to be yellow. And we know that under the black is also yellow. So this is an all sugar fermenter. It uses glucose and at least one disaccharide. And it's positive for H2S. If we look at Proteus mirabilis, the slant is pink, but the bottom is black. Again, underneath the black, we know has to be yellow. So this organism is a glucose-only fermenter. It only utilized the glucose, right? We know that it's yellow underneath. It did use the glucose, but it did not use a disaccharide. And this organism is positive for sulfur reduction. It did produce H2S because we see the black. Here is our alkaligenase faecalis. It is pink throughout. So this would be an organism that is non-saccharolytic. It did not use the sugar. It only used the proteins, and that's why it's pink throughout. If we look at Pseudomonas aeruginosa, notice that the tube is pink throughout. Now, you might notice something interesting here, that this top part here is black. Notice it's not in the butt, it's like on the very top. That is actually not positive for H2S. It's not positive for H2S. That black that you're seeing is actually a pigment that it's producing, but it's not utilizing the hydrogen sulfide. It's not producing H2S. So this organism would be, in this case, non-saccharolytic. It didn't use the sugar. It only used the peptones. Then we get to E. coli. E. coli is yellow throughout. And notice that it broke the agar. The agar separated. That's because E. coli can use glucose and at least one disaccharide. That's why the slant stayed yellow. It used at least one disaccharide. And it does not produce H2S. It's not going to produce hydrogen sulfide because there's no black, but it did produce gas as part of its fermentation. And so the yellow is an all sugar fermenter. It's able to use glucose and at least one disaccharide, but it is negative for H2S. So in this experiment, we have 3 that are positive for H2S: Salmonella coloraceus, Proteus vulgaris, and Proteus mirabilis. The other 4 are negative for H2S, but have some variability in terms of which sugars they use or do they not use sugar at all. Now, for this test, you don't have to memorize which bacteria gave which reaction. I'm not going to ask you that. You don't need to memorize that. But if shown one of these tubes, you should be able to tell me, what does that tell you? What is your interpretation? Did it use glucose? Did it use a disaccharide? Did it produce H2S? You have to understand what the different colors mean. And so my recommendation for this one, this is one of the tests that's a little bit trickier. So what I would recommend for this is to find a partner and explain these. This is what I do with students in class, is I have them explain this to each other. And without looking at the interpretation, like use this slide and see if you can explain to your partner why these different tubes are the color that they are. What does that tell you about that organism? And so practice that while looking at these tubes. And so this is going to conclude our triple sugar iron agar test, our TSI test.